Yes, hi everybody. I'm Indira and this is Gianna. And both of us, we are postdocs based in the Robert Gordon University in Aberdeen. And today we are going to talk a little bit about the project that we're working on, which is funded by the Natural Environment Research Council. And it's about eliminating harmful algal blooms in salmon farms. Did it change that slide? Yes, perfect. Please just unmute yourselves and let me know if there's something like you cannot see the slide properly or something, and feel free to just drop a message or interrupt. But yeah, so I would like to start giving you guys a little bit of a background about the project and how the idea kind of like came up. So initially, salmon was considered to be like very fancy food and like very luxury like food item, but nowadays we can see that it's becoming more and more common and like more present like in a daily basis type of thing. So nowadays around 1 million salmon meals are consumed per day in the UK, and we can also see the growth in the salmon industry when we consider all the like more statistical type of data. So for example, last year, Scottish salmon, which is the name of farmed salmon here in Scotland, it was named the UK top food export. And another way of like seeing this growth in the salmon industry, in the Scottish salmon production, is when we compare the number of salmon farms 50 years ago, which were around like just a couple of them, just a couple of farms, in comparison with nowadays, which we have around 200 of them. So it's a massive growth. But unfortunately, the farmed salmon, the Scottish salmon, they might face a few challenges. And one of them is the presence of harmful algal blooms in the salmon farms, which might have a negative impact in the production of the fish, of the salmon. And this might be due to the mortality of the fish because of the algal toxins that might be present in these blooms. It could also be because the anoxia that would come from the high density of these algae, the high biomass that is present in the bloom. And it could also be because of like some physical irritation that can be caused in the gills of the salmon. And so here I'm just bringing a few examples of some incidents that happened in different salmon farms. So a more recent one happened last year in 2024, the beginning of the year, in Chile, where 5,000 tons of salmon were killed because of harmful blooms. And we also have some examples in the UK. So for example, in 2019, there were some Scottish salmon deaths because of harmful algal blooms as well. And then right after that, we had a similar incident in Norway, also caused by these harmful algal blooms. So obviously, this is like environmental impact. It causes like issues with like the concentration of oxygen and the death of fish and the presence of harmful algae blooms. But this also represents an economical issue because, as we saw before, it's an item that is being consumed more and more every day in the UK, and then the death of this fish then consequently cause some financial issues as well. So because of this, we need to find ways to overcome this problem and the impact that the harmful algal blooms cause in the salmon farms. And one way of doing this is to using approaches like targeted approaches that won't cause damage to the salmon, but then we'll be able to remove these harmful blooms from the salmon farms. So one way of doing this is to use photocatalysis, which is an advanced oxidation process that is based on the generation of radicals. And these radicals that have high oxidative power, and they are able to promote the degradation of both the cells of the harmful algal blooms, but also the toxins that they might produce. And for this technology, for the photocatalysis to happen, there are two main things that are involved and two things that we need, which are one, the catalyst, and two, the light energy or the light source that we need to activate the catalyst. And then the two of them together would ultimately produce the radicals that I just mentioned. So when we go in more detail on the light source, usually UV is the mainly used type of light energy that we need to activate the catalyst. However, the UV can be separated in three different bands, which are the UVA, the UVB, and the UVC. The UVB and the UVC are the most known bands or types of UV that we can find because they are known to be more in the disinfectant level of UV. However, they can be harmful. They can cause cancer and they can damage cells. So they are not ideal. However, we also have the third type of UV, which is in the UVA band. And this is no longer in the disinfectant band or like the harmful radiation level of the UV, because it's more closer to the visible light range, and it's very close to the blue light spectrum as well. So this will be better because it will also be able to activate the catalyst during the photocatalysis and the treatment. The issue with the UV, another issue with the UV is that we can also have some issues with the cost of the UV radiation. So the UV also help us with this because it's a low energy type of light, but the high cost associated with the irradiation also need to be considered. And one way to overcome this is to use light emitting diodes or LEDs, because they are not only just affordable. For example, nowadays we can find one LED for less than 1p, so that they can be quite cheap, but they also have long lifespan. So, for example, LEDs are supposed to last at least 10 years. They can also be very easily replaced, and they also require lower energy input, and they can easily be integrated into a reactor design, for example, and also they can be easily waterproof. So these would help when we're considering the environmental application of the technology. So the other thing that we need to consider, as I mentioned before, is the catalyst that we're going to use. So initially, when photocatalysis was first studied, the catalyst in the powdered form was usually applied because it has a great surface area, and this would translate in a high photocatalytic efficiency, which is obviously what we want. However, because it's in the powdered form, it could be dangerous if inhaled, and then it would be hard to remove it from the water, for example, like to actually physically remove, because obviously it's a very fine powder. And instead of like promoting the degradation of contaminants, it would actually be adding powder of like a specific chemical to the environment, and obviously we don't want that. So the removal of the recovery of the catalyst would also be a challenge. Then some catalysts in the pellet form started to appear, and obviously now, because they are bigger than the fine powder, it will be easy to remove and to recover after the treatment. And because we can recover, we can also reuse this for like different cycles of photocatalysis. And it still has a good surface area. The only drawback is that it has a lower photocatalytic activity in comparison with the powdered form of the catalyst. So the most recent idea was to use different materials as immobilization matrices to coat the powdered catalyst form onto the surface of these materials. And then because of this, now we wouldn't have the powder aspect of it anymore, so we'll be able to eliminate the dust. And the fabrication, the coating procedure is cheap and easy. And then because now, again, we don't have the powder anymore, we have something bigger in the scale of like 2 to 5 millimeters, we can recover the catalyst after treatment. And again, we can reuse this for different cycles. However, the only thing is that we need to consider during the coating process, it needs to be very stable. Otherwise, we can have some shedding of the catalyst. And then again, we'll be adding some of this loose powder into the environment. And obviously, we don't want that. So the idea that we had was to no longer use the powder catalyst form, because again, when we're considering large-scale applications in the environmental application of technology, we cannot just be adding this to the powder to the environment. So we decided to use glass beads as our immobilization matrix. And these glass beads, they are good because they are made of recycled glass, which brings on the sustainability aspect of the project. And it's also very affordable because these glass beads, they are used in construction companies for thermal insulation, and they can also absorb sound. And so we'll kind of be repurposing this type of material. So then we made 2 different types of catalysts with these beads. The first one was the titanium dioxide coated beads, which is the white bead on the right-hand side. And then we also made graphite carbonitride coated beads, which is the yellow beads on the right-hand side. So the first opportunity that we had to apply this type of technology and the coated beads into a more environmentally relevant project was this pilot study that was done in Brazil. And this was done in this big drinking water reservoir that is known for having perennial cyanobacterial blooms, which is one type of harmful algal blooms. And that's why here on the right-hand side, we can see the green coloration of the water, like the reservoir water. So the idea of the project was to use these coated glass beads and then put them inside of pods. And then close to these pods, we are going to use UV LEDs to activate the catalyst. Then we would place the system, like these reactors, inside of the reservoir close to cyanobacterial blooms. And then this will promote the radicals to then degrade the cyanobacterial cells and also the possible toxins that they would produce. So here, this is just a schematic of what happened in the reservoir. So on the left-hand side here, I don't know if you guys can see my pointer, but on the left-hand side, we have the representation of the reactor that was used in the experiment. So this consisted of a cylindrical reactor made of a stainless steel mesh. Then inside of the cylinder, we would have tetrahedral pods containing the titanium dioxide coated beads. And then on the wall of the cylindrical reactor, we would have UV LED strips that would be able to activate the catalyst. Then we place 3 reactors inside of a mesocosmos bag, which contain around 3,000 liters of the reservoir water. And then on the right-hand side, you can see the top view of the platform containing the mesocosmos bag that was used in the reservoir. So we had 3 mesocosmos bags containing the 3 reactors, which consisted of the photocatalytic treatment. And then we also had 3 other mesocosms with no treatment, which worked as our control. And the study was supposed to last for 6 weeks. However, halfway through, the experiment had to be interrupted because apparently some piranhas decided to come and join the party. And then they ended up biting the mesocosmos bag and making holes on them. So even though the experiment was interrupted, we could still see some promising results. So for example, on the left-hand side, We can see the difference in the color of treated samples to untreated samples within only 72 hours of treatment. And overall, by the end of the experiment, we were able to see an overall improvement of the water quality. We could also see a decrease in the cyanobacterial cell density, and also a reduction in the trichome length of some cyanobacterial species.
Okay. Sorry. I'll take over from here. So thank you, Indira. So after the successful first trial in situ I've done in Brazil, we decided to try the same technology, but in a different environment. So this time, instead of a freshwater reservoir like it was done in Brazil, we decided to go for the marine environment and to go for different applications. For the idea of this other project, it was to protect shellfish farms with a photocatalytic curtain. Instead of having the coated beads activated by UV in a pod like it was in the previous project, for this one it would be a curtain and then would activate using UV. And because it was a different environment, usually different organisms are the problematic. So instead of cyanobacteria for this other project, for the shellfish project, the model organism was Alexandrocatanella. Alexandrocatanella is a marine phytoplankton, is a dinoflagellate. It's usually occur in a single cell or like in very short chains, up to 8 cells. The problematic of these organisms is because they can produce saxitoxin, also known as paralytic shellfish poison. So if the shellfish is exposed to this type of toxins and consumed by humans, it can give some kind of health issues. So this have been a problem to the shellfish industry. So in order to know if the idea of the project is actually feasible, we've done a kind of a larger bench scale experiment with Alexandrocatanella here in our facilities. This bench scale experiment contains a fish tank with a photocatalytic curtain activated by the UV. The difference is we had two different types of curtains. We had the titanium dioxide curtain, and we also test a carbon nitride curtain. There was no muscles at this point of the experiment. We only evaluated how the Alexandrocatanella cells would react with this photocatalytic curtain. And we took samples over 24 hours. So during the 24 hours, we could see that both the photocatalytic curtain forms, the titanium dioxide and carbon nitride, they were very efficient on removing the Alexandrocatanella cells. Within 3 hours of experiment, approximately 60% of the Alexandrocatanella cells were removed, and we reached very close to 100% in 1 day of experiment. So after these very promising results, we decided to go larger, but instead of using the titanium dioxide coated beads, we went with the carbon nitride coated beads, just because the carbon nitride coated beads, they are easier to coat, they are easier to produce in our laboratories. So we performed like a pilot scale photocatalytic curtain experiment back in Cephas, up over there in England. So for these ones, there was basically in a tank trial, they would recirculate Alexandrocatanella, simulating a bloom of Alexandrocatanella. For this experiment, we evaluated the muscles instead of the Alexandrocatanella cells. We had 6 different setups. We had a positive control, which was basically exposing the muscles to a simulated like Alexandrocatanella bloom, without any barrier, without any protection. We had negative control. For this one, there was these tanks, they were not fed with Alexandrocatanella cells. They were just recirculated with seawater. We had a curtain-only setup, which was a curtain, but instead of having carbon nitride coated glass beads, we only had uncoated glass beads. So the aim of this setup was to evaluate if the curtains would have any physical removal of the cells. We had a setup that was carbon nitride coated beads activated by UV. We had the same curtains with carbon nitride coated glass beads, but this time without any UV activation. So there was no lights from the UV, and we had the UV only. So for these experiments, as I said, the Alexander Catanella cells, they were not evaluated. What we did was we evaluated the intake of the muscles of the toxins that the Alexander Catanella produces, and we sample the muscles after 24 hours contact, 48 hours contact, and 72 hours contact. So the results, they were quite promising. So in these graphs, I just want to highlight that the graphs, they are purple. It just means they were the graphs that had the UV lights on. So the positive control, it was the tanks where the shellfish that had absolutely no protection from the Alexander Catanella blooms. So that means that, as expected, they have the highest intake of the toxins. We noticed that the toxins that were most commonly found in the shellfish were neotoxin, saxitoxin, and GTX4, which were saxitoxins variants, and they are very harmful. And especially they can bring food poisoning to humans if the shellfish is ingested with these toxins. So the positive control had the highest concentrations of these Alexander Catanella toxins. The negative control, as we expected, we had negligible intake of the shellfish toxins for the shellfish. The curtain control, we could see a slight remove of the intake of the toxins by the shellfish. So that means that the curtain, they also act as a physical barrier for the Alexander Catanella cells. But the main promising results, they were in the treatment. So the carbon nitride coated beads activated by the UV lights, they had a very significant reduce and decrease of the toxin intake by the shellfish. The carbon nitride only also had a very promising removal of the toxins, and also the UV only also had like a very significant decrease of the intake of the muscles of the toxins by the muscles. So after these experiments, we decided to go further and use the same type of idea, the same type of setup to different applications and different environments. So that's where we took the idea of eliminating harmful algae blooms in salmon farms. Is also more environmentally relevant for the Scottish industry. But because of the volume of the water, because usually salmon farms, they are like in ocean, we needed to do some kind of adaptations that it's not possible to treat like a whole ocean. So the idea of this project is to combine with the photocatalytic curtains, combine the lure lights, which would attract the target harmful algae to the reactors, and then only then remove these harmful algae blooms from the environment. So this idea came from this publication that found that depending of the light, depending of the wavelength, and depending of the intensity of the lights, and also depending of the organism, different organisms would be attracted to different types of lights and wavelengths. So the main goal of the project was to deploy in very close to salmon farms cages a reactor that would have a combination of photocatalytic curtain and the lure lights, and then only attract the harmful algae without affecting the rest of the biota, not attracting the type of microalgae or microorganism. And only then when these harmful algae will attract to the reactors to start the degradation process. So this project is a partnership with SAMS, which is the Scottish Association for Marine Science. They are the ones responsible for the study and for the evaluation and development of the rule-lights. They would kindly agree for me to share these results that they perform over there. So that's a very small scale experiment with marine dinoflagellate. There we can we see them going towards the light. So they are very much attracted to the blue and green wavelength of LEDs. But because, as I said, that's a target approach, they also evaluated that depending of the organism, they would react differently to the same type of wavelength. So we can see here on the left-hand side that this green microalgae, instead of going towards the light, they start like spiraling, whereas the one that I showed previously, they go towards the lights, they go attract to the lights. So because of these characteristics of the microorganisms, we can select the type of algae that we would like to attract to our reactors. So that is another experiment performed over there. So that's a larger scale experiment. So it was performed in a fish tank, and it's the same type of microorganism, which is Scriptella, the one that was moving towards the light. And of course, because it's a large scale, we don't see as clearly and it's as obvious the microalgae going toward the lights. But if you bear with me, you're gonna see it very soon. Here on the bottom, that's where the Scriptella start concentration, forming this bloom, and then very soon you're gonna see very obvious layer going up towards the blue light. So that's where you should focus your your sight, and then very soon you're gonna see the layer going up. Like the faster we play the video, more obvious we can see the microalgae going up. I'm gonna help you out very soon, but like we can see here the layer already forming and the layer already going up. If I fast forward the video, and when I do this, we can see much more clear the microalgae going towards the light. So, because that's the marine environment, and the salmon farms also struggle with Alexandrocattanella's blooms. It's not only the shellfish industry. We wanted to use the Alexandrocattanella as a model organism, but the problematic is Alexandrocattanella, they don't form very dense blooms, and they are very difficult to scale up in the lab. So while we are cracking this code, while we are trying to make this happen, we decided to go forward with a cyanobacteria that can, depending of the strain, they produce the same type of toxins as Alexandrocattanella, like the very similar type of toxins. And this one is a very fast grower, and we have a very consistent, reliable culture system here in our facilities. So we decided to have like a proof of concept and a bench scale experiment here in our facilities with Rhaphidiopsis raceboscis. That produces PSP toxins. So for this one, we can see here in this spot that in 1 month we had a very dense culture of this cyanobacteria. So we performed this experiment using natural water. We decided to remove all the media that this cyanobacteria grow. Indira is going to publish very soon a publication that shows that depending of the media, the nutrients can affect how the photocatalytic activity is going to happen, and we want to have like the closest as possible environment relevant scenario. So we removed the media, we added natural water, and we left these organisms sitting for a whole weekend just to let them acclimate to the experiment conditions. And then these cyanobacteria, they were exposed to a light control, which was LEDs that could emit UV and visible light. They were exposed to the treatment, which was carbon nitride coated beads activated by the same LED lights. And we had a dark control, which was the carbon nitride coated pits, but this time without any irradiation by the UV LEDs. So in 24 hours, it was very obvious to see that the treatment had completely degraded the cyanobacteria. It was only at 72 hours that the UV light by itself showed like very clear degradation or very clear impact on the cyanobacteria. But it also at the same time was when the dark controls started like struggling a little bit. So we also evaluated the photosynthetic activity of the cyanobacteria along the experiments. And as we expected, in 24 hours, we couldn't detect any photosynthetic activity. So I just want to I wanted to highlight that this dashed red line over here means that anything below this is showing photosynthetic distress or damage to the cells. So within 3 hours, both the treatment and the UV control showed stress to the cells of the cyanobacteria that we analyzed. The cell density, as we saw in the pictures, within 24 hours, there was no cyanobacteria cells present in the treatment anymore, whereas for the UV control, it only happened at 72 hours. So here on the left-hand side, we can see how dense were the cyanobacteria that we selected to perform this test before we start the experiment, and how it looked like under the microscope after 24 hours exposure to our treatment. So for the future research, is we have established and we have supported with our data the photocatalysis work, and also the law of the microalgae also work, depending of the microalgae and depending of the lights used. And from now on, we are going to focus on this project on combining these two technologies to build and develop a reactor that can be deployed in salmon farms that have both the photocatalytic removal, but targeting the harmful algae with loading. Thank you very much. If you have any questions.